Something a little different today. Um, I thought I'd do a little scientific paper review for you in case you're interested. The paper I'd like to discuss is the following one that should appear on your screen. And it's related to tattoo ink and its impact on the immune system. So this popped up on my radar because I listened to Brett Weinstein's Dark Horse podcast, where they presented this paper very briefly, and I'm obviously not going to criticize what they said, but it kind of sparked my interest because I hadn't read the paper. The podcast came out, I think, a couple of weeks ago. The story also broke mainstream media, popped up in the LA Times. It was picked up by the Global Vaccine Alliance, Gavi. And so I thought it's kind of mainstream enough for me to actually look at the paper, which was published in PNAS, which is a really good journal, Proceedings of the National Academy of Science in the US. And well, obviously I'm an immunologist, and my skin contains tattoo ink, so I thought might as well look at what the latest results show. And so I was intrigued by the few things that were presented in the Dark Horse podcast, and I decided to have a look myself and give you my take on it. So let's start with the beginning, and I'm gonna put my immunological reviewer hat on. If I had a manuscript like that, obviously unpublished yet, I would immediately realize that the title is very non-committal. Like, the title is Tattoo Ink Induces Inflammation in the Draining Lymph Node and Alters the Immune Response to Vaccination. So non-committal, alters, like, so you're basically not saying which way it goes. It doesn't dampen, it doesn't enhance. So alters already is quite descriptive. So they don't really want to make a point. And induces inflammation. Inflammation, it's such a vague terminology for immunologists. Inflammation doesn't mean anything. So as soon as things are non-committal in the title and you actually have to dig deeper in the abstract, you know that it's not a clear-cut story whatsoever. You can already tell that from the title. That's the title. I mean, it's a great title. I would have not used a different title if I'd be responsible for that work, because again, the data is not very clear-cut, as I'll present to you. So I mean, there's already so the introduction sets the scene. There's a bit of literature of tattoo ink, which can be found, which, like, you would assume it spreads throughout the body. There's no doubt about that. In humans, it can be found in the liver, taken up by liver macrophages, also called Kupffer cells. And long term, you can detect it throughout the body. The question that these authors wanted to address is, well, what does it actually do? Is there any kind of issue? Is there a toxicity profile with regards to the immune system, and just to see what it does. So the work was predominantly done in mice. They used some human cell culture systems, which again they're very crude. It doesn't tell you exactly what's going to happen in a human whatsoever, but it kind of gives you a little bit of hint if there's a signal there to follow up in humans. But the paper was done in mice, and so just quickly some of the results. So they tracked the dye in the draining lymph node. So if you inject anything in any parts of your body, the lymph will bring whatever you injected to the draining lymph node. That typically from an immune perspective is exactly what you want, right? Because in the lymph node, that's where the immune action happens that will then be responsible for the specific anatomical site. Now, of course, if you tattoo, typically also hit a few veins or little arteries, little capillaries. So if it reaches the blood, it will obviously circulate systemically throughout the entire body as soon as it reaches the bloodstream. That makes sense. But if it's really only in the interstitial space, that's taken care of by the lymphatics, and the lymph will then drain some of the ink to the draining lymph node. And the lymph node is has a very complex architecture that makes sure that the proper immune cells are being shown what's happening in the tissue. So if there's anything that needs to be reacted against, that action is going to take place in the lymph node. So they detect the ink in the draining lymph node, and then they they wanted to know, well, who picks up the ink, and unsurprisingly, it's macrophages. So they predominantly find the ink in macrophages. That's not surprising either. Macrophages are kind of the garbage bins. They are phagocytes, typical phagocytes. So they take up particulate matter, and they tend to digest it, or process it. In the case of an antigen, it will also present antigen to cells to say, okay, this is what I picked up. Maybe you should have a second look at that. In the case of ink, it's not really being presented. It has to be cleared. It's kind of a chemical agent, so it will try to digest it enzymatically. So that's not surprising that the phagocyte would take that up. The first kind of hint towards an immunological effect that might be classified as bad, although there's no bad, good in the immune system. Reactions just happen, and then you just figure out what the consequence of that is. So the first kind of consequence that they describe is apoptosis. So the phagocyte, the macrophages that take up the ink, they die. So apoptosis is a regulated cell death system, well, if you take it directly, of course, if you perhaps not really trained in immunity, you say, well, cells are dying, so that's horrible, that's a toxic effect. We need to put that into context. So macrophages can very often apoptose or die when they ingest something that they cannot really digest. So like, imagine like they take up something, they try to digest it like a human would do, but imagine like you ate something, and then basically it's like a stone, and you're not going to get rid of it. Well, as a cell, what you're going to do then is apoptose, so create a death, and then the entire cell will kind of disassemble and will then be being able to be picked up in smaller pieces by other cells. So this is a regulated system, happens, I would say, fairly often. Typical example would be alveolar macrophages in the lung. So if you breathe in dust, if you breathe in any kind of particulate matter, you have alveolar macrophages. So they sit not inside your body, they sit literally technically outside the body. So you have the lung epithelium, and so they sit outside. They sit at the air interface. And they pick up things if you inhale something or smoke or anything like that. These cells, will pick that particulate matter up, and they are renowned to die immediately when they're exposed to something that they shouldn't be. And then they're getting renewed. So there's constantly new macrophages coming up and doing the same thing. So it's almost like a first line of defense. So detecting some apoptosis or cell death by macrophages is not really concerning. These cells are being repleted all the time. I mean, that's almost what their job is. Now, I don't want to play too much devil's advocate here. Of course, if it seems to be toxic to the macrophage, so it seems to be something they cannot just readily enzymatically get rid of. So yes, and that makes complete sense. It's a tattoo ink it's a dye, so it's a chemical. It's not something that the body can easily get rid of. So yes, so there is apoptosis of these macrophages. Then they did some in vitro culture work with human macrophages. So these are not primary macrophages, but macrophages they artificially derive from human monocytes. So you take monocytes from the blood and then you give them some cytokine cocktail. And then they differentiate in cell culture to what we would say as monocyte-derived macrophages, not primary macrophages. Sorry, and they obviously take up the dye too, and there was also apoptosis detected. Now, interestingly, and this is this, you know, I've done mouse work in my immunological career, and I've done also human work, and the immunologists that only do almost exclusively do mouse work, and then there's human immunologists. And there's a reason for that, because the systems are not exactly the same. Like one of the caveats already, if you do something in the murine skin, so in the mouse skin and the human skin are immunologically distinct. So there are cell types in the mouse skin that don't exist in the human. And so, and also in terms of thickness and architecture, it's not exactly the same thing. Now, this here we're talking about tattooing, so they're more interested in what goes to the draining lymph node. But just as a reminder, like the skin is not exactly immunologically identical in humans and mice. But here, in terms of cell culture of the human, they detected some apoptosis and some toxicity, but it was also very ink specific or ink color specific. They used different ink colors, so black, red, and green. Traditionally used, black is probably the most used in tattooing. But here again, the murine, the mouse macrophages and the human macrophages displayed a different behavior with respect to those different inks. So there is already a human mouse difference, which is completely expected. Like I would have not expected nothing else. We know that the cells behave differently. But there is a toxicity signal, so I'm not denying that whatsoever. So, but again, macrophages do that. Not concerning. It's like quite expected. Where I can already start, again, putting my reviewer hat on, as soon as you're going in vitro, and that's true for a lot of immunological work, how are you going to define the concentration of the ink in cell culture? We always talk about physiological relevance. So were those physiologically relevant concentrations? So if you want to work in vitro, ideally you would have to take out the lymph node of an organism and quantify the amount of ink so that you can reproduce those concentrations in vitro. Otherwise, in vitro, what you do is, they use two different concentrations. I'm not sure what they mean. I'm not sure if they're high or if they're higher than what you would see in the draining lymph node of a human or mouse that has been tattooed, or if they're lower. So there's a bit of lack of relevance here, or they should have discussed it a bit clearer how they came to those concentrations. But that being said, they find a signal, which is fine. And so So for now, expected ink drains to the lymph node. In the lymph node, the phagocytes that usually do that job take up the ink, and there is some cell death happening. Now let's come to the inflammation part, which is in the title, right? Tattoo ink induces inflammation in the draining lymph node. Again, in the title, very vague. I would have not known what they actually did. You have no idea what they've done in the draining lymph node to say that there's inflammation. So if you have that amount of that, or that level of vagueness, you know that it's not black and white, as I said previously. So inflammation here, they looked at immune cell recruitment. So as I mentioned previously, the draining lymph node is a complex structure, and if something is happening in the tissue, the draining lymph node will get wind of it. Like some cells might come to the tissue, from the tissue, go into the lymph node and say, guys, there's something happening there. And then depending on what's happening or what the signals that are being given are, you might want to call for backup. So then an immune reaction takes place in the draining lymph node that might attract other cells to come into that lymph node to help with whatever is happening. And so what they detect is an immune cell recruitment to the lymph node. The lymph node, as you might know, if you have an infection, the lymph node gets bigger. That's a GP would look at the lymph node, see if it's enlarged. And so the enlargement is because other cells are being drawn to the lymph node, are being called to the lymph node through chemokines, different signals that tell the cell, you need to come here, you need to help out. So they see immune cell recruitment, typical cells like the dendritic cells, which are antigen-presenting cells. You have NK cells, T cells, B cells, all the immune cells that typically do a specific job, an important job in immune reaction. So the immune recruitment by itself is, it could be called inflammation. I mean, there's something happening. So again, inflammation is a very vague term. So yeah, there's a reaction, a recruitment of immune cells. So you might call this inflammation. That's fine. The recruitment is fairly acute, so it happens very quickly, and is still prolonged after. So what I really appreciate in that paper is that those are big experiments in mice. So because they extended the observation period to 2 months. So 2 months is actually a very, very long experiment for mouse. For my mouse experiments, 2 months is a long period to take care of the mice that have been tattooed in this case. So there is prolonged recruitment of immune cells. So, and also that matches really well with what they've seen, that they can detect the tattoo ink 2 months down the line. So this is one of the if I would have had to guess without looking at that paper how long I would have assumed the ink to hang around, I'm not sure I would have said 2 months. I would have potentially said that it's cleared earlier, and it might actually hang around longer because they only observed the thing for 2 months, right? So it could be that it hangs around for much longer, which that's definitely a concern. I would love to know for how long that actually is the case. So because the thing that bothers the immune system is still there after 2 months, it makes sense that there's still some activity in the draining lymph node. Now, to sustain or to support that claim of inflammation, they measured also some cytokines and chemokines. So chemokines are chemoattractants. So chemokines typically, these are the signals to tell immune cells to come in. So if chemokines are up, you will recruit immune cells constantly. And cytokines are inflammatory mediators, like typical ones, IL-6, TNF. So IL-6, TNF, these are quite acute cytokines. So that's what they've seen within the first day of tattooing. you had an up in the cytokines, and then later on you see quite interesting cytokines like IL-1, IL-1 beta, which definitely is kind of a concern. That's kind of a quite potent inflammatory mediator. But also to put that into perspective, so not really talking about the cytokines, but the immune cell recruitment, they've also seen immune cell recruitment with, so that control was to tattoo, to do the tattooing procedure just with saline. So they just basically punch saline into the skin. So that was almost that control for the trauma to the skin. And just that control, just inducing the tattoo trauma to the skin, that also increased immune cell recruitment to the draining lymph node compared to not tattooing at all. Now the signal wasn't as strong as obviously tattooing with ink, but there was still a signal there, right? So part of also what contributes to the injury and to the activation of the entire system is also the fact that you're injuring the skin. It's also probably important if you're doing things in animals. I'll put a picture up of how it actually looks like when they tattoo the foot pad of the mouse. Proportionally, it would be interesting to kind of have a comparison of what what kind of surface are we talking about in humans? What would be the equivalent of percentage of total body surface that's being tattooed? Because that would actually be quite a significant percentage, I think, if you're looking at like the surface that they tattooed on those mice. It would be, it's hard to give a correlate to humans, how much that would correspond to. Definitely relevant. I mean, there's people who tattoo an entire arm, so that probably the equivalent of that. So also if you thinking about having a small little tattoo, I don't think this data would be nearly as relevant because the dose and the concentration would be much, much lower than what they've done here in animals. Also, just to put that into context. So they've seen cytokines in the draining lymph node, they've seen immune cell recruitment. So there's definitely something happening. So the next step, absolutely, which makes sense, is to look, okay, so we've observed these changes. So there's immune cell recruitment, there's cytokine release. Okay, what does it mean? So the next step is to logically do something that means something, instead of just saying, well, there's more cells and there's dye, there's still ink there, and there's these cytokines. So what they've done is to give those mice that received tattoos vaccines. Now this is fairly smart because you can then administer the vaccine basically where you tattooed, so that it drains to the same lymph node. So the lymph node where you already checked, what's the functional consequence of the immune cell recruitment and the cytokines that you've seen? Does it alter then that lymph node when it receives an actual immunological threat? I mean, like the ink might also be seen as a threat, but like a real robust task to do, which is to respond to an antigen that's given via a vaccine. So they used 2 different vaccines. That was a nice confirmation. They gave the Pfizer BioNTech COVID vaccine, and then they also used a simplified inactivated influenza vaccine. So 2 different vaccines. So I'll show some pictures here. So basically what they observed, and that's exactly looking at the results, it explains why the title was so non-committal, because they had two different observations with the two different vaccines. So, and so they probably done that in that order. So first they started um, with the COVID one, and then so what they saw is a decrease in antibody titers. So meaning that you're getting a tattoo somewhere, like let's assume you get a tattoo on your arm, and then you get the COVID shot in the same arm, drain kind of anatomically like in a similar site, so it drains to the same lymph node. You get less antibody production to COVID, to the COVID vaccine, as you can see. There is some, as with most of what they looked at, there is some variability depending on which ink you use. You know, again, black versus red versus green. And they also checked, and that's actually quite neat. So they checked the two different time points after the tattooing. So 2 days afterwards, where you assume there's still kind of inflammation locally, you know, in the skin and everything like that, but also 2 months. So once the tattoo has arguably completely healed and everything is at baseline, except for the fact that at 2 months you can still detect some ink in the lymph node, you can detect immune cell recruitment, but the skin would be healed, right? So they do it 2 days and to compare 2 months after the tattooing. And in both cases, they see reductions. So it's not really that, well, 2 days, yeah, well, there was still kind of inflammation on the skin at least. There was some wounding. No, if you actually, the skin is fully healed, you still see the effect, which again matches what they see in the draining lymph node, that you still see something there. I would assume, I'm not sure whether the authors would have been surprised at that stage, because again, you have more cells. Okay, macrophages die off, but you have more cells in the draining lymph node. You still have the ink lying around, and you have pro-inflammatory cytokines. So potentially you could have argued that, well, maybe you see more action, right? Because there's some kind of inflammation going on. So they see reduced antibody production. So the immune cell seems to be dampened. And so I appreciate what they've done next is to actually look at mechanism. Okay, how do we explain that it's less? And so they're looking at, as shown here, they're looking at different co-stimulatory molecules. So CD, as you can see here, CD86, CD80, and MHC class 2. So CD80, 86 are, this is what an antigen presenting cell, like a dendritic cell or a macrophage, require. So it's like a signal giving to a T cell to say, this is what I'm presenting to you. And by the way, it's a threat. So you can't just present antigen, because then the T cell says, well, you're presenting this thing to me, like you also need to tell me that it's a threat. So these molecules, if they are lower, they're going to tell, well, the T cell, okay, I found something, but, and then be a bit ambiguous, like how much of a threat is it really? So the fact that CD86 in this case is lower, it gives less of a signal like, oh, this is a threat. So it kind of makes sense. So if those co-stimulatory molecules are down, I mean 86 at least, 80 not so much, or 80 at 2 months, and 86 at 2 days, so 48 hours. So if those are down, it makes mechanistic sense that, well, you would have less of an antibody production going on because the threat signal was lowered. So that makes sense. Now they also looked at the antigen, so spike expression on the macrophages. Remember, the macrophages are the ones that take up the ink, and they might be influenced by it because it's slightly toxic. So there is less antibody, or sorry, antigen presented by the macrophage. Now I understand why they did that, right? Because the signal of the ink or the ink uptake was done predominantly by macrophages. So you would want to see what the macrophage look like phenotypically, or how much antigen it presents, as well as the co-stimulatory molecules. But what slightly bothers me is I would have loved to see that same thing done on dendritic cells, because dendritic cells are actually the main professional antigen presenting cell in a case like that. So if you forget about the tattooing, forget about the ink, if you would look at the vaccination response in the draining lymph node, you would probably not predominantly look at macrophages. You wouldn't. You would look at dendritic cells, 100% sure. So the fact that they look at that is because of the ink. But you cannot just look at what is impacted by the ink. You also need to look at what you typically look at when you do looking at vaccination responses in the draining lymph node, which would be DCs. So yeah, I would have wished that to be a bit more clear, but that's fine. So they see a reduction. So at this stage, I understand that they would have looked at a different vaccine, at another vaccine, because imagine you're at that stage in your project. So you tattoo the skin of mice, you see inflammation, immune cell recruitment, but then you have these kind of somewhat conflicting decreases in antibody production. So you tell me, okay, it's inflamed, but then it's suppressed at the same time. So you kind of, there's something missing, right? It's very observational and very non-committal. So I appreciate that the authors gave it another shot with a different vaccine to see maybe it's just because of this mRNA vaccine instead of another vaccine, a more traditional one. So they use the inactivated influenza vaccine, and there they see an increase in antibody production, as you can see here. Again, the statistical significance is dependent on the ink or the ink color that they used. And then, I mean, that they basically left it at that. So that reflects perfectly the title. You know, they detected inflammation in the draining lymph node, and it alters the immune response to vaccination. Alters because it's vaccine specific. It alters it. Sometimes it goes up, and sometimes it goes down. So it's very, very difficult to frame that result or what it actually means. I mean, my take-home messages and how I would frame this research for myself and for you, if you're interested in these kind of things and you happen to carry some ink in your skin. Big take-home messages is it's very specific for the type of ink. So the color. And because of the color would obviously represent a different chemical structure. I'm not sure how they would derive those inks. So these results are very specific to what kind of tattoo you would have, whether it's in color, whether it's black only, and where you got it. There's country-specific regulations on inks, and I definitely agree that the results are compelling enough to warrant some kind of regulation of what is being put in those inks. If those substances really hang around the bodies for so long, I think there needs to be certainly some decent regulation around it, and some countries are certainly more regulated than others in that regard. So it's really reframe this in the context of, okay, what kind of inks has been used, because it might be that some inks have very limited impact, and some inks have more of an impact, and it might not even be related to the color. It could be very you might have a black ink used in one country and another product, also black ink, in a different country, and because of the extraction, because of the manufacturing process, one might be 10 times more toxic for macrophage than others, or it might induce something else. So it's very, very specific in this context. Again, the proportionality. If you have a tiny little tattoo somewhere, which might actually spread to different lymph nodes, the impact might actually be very, very small. If you have a large area of your body tattooed and a lot in black, there would be much more of an impact to that, depending if the black ink that has been used also displays those kind of characteristics. What else did I wanted to say? What's interesting, so the authors then in the discussion frame this in, well, there's something happening in the draining lymph node, and the impact of what's happening will have different consequences on different vaccines because the mechanisms of action of the vaccines are different. Now, this is a very important point. And this is exactly when I discuss vaccines actually more in detail and vaccine side effects. This is exactly why vaccines should never be bunched together. There might be very safe vaccines, and there might be vaccines, like for instance an mRNA, that we mechanistically understand far less. The risk and side effect profile of vaccines are very vaccine specific. And this is a perfect example, right? You have two different mechanistically different vaccines, one traditional, the inactivated flu one, and then you have the mRNA, the new one where we don't know much about it immunologically. So something like a tattoo experiment basically teases apart mechanistically those two vaccines, because then the result will be different. So I think that's a very important point. It's almost like magnifying these mechanisms of action of those two vaccines is completely different, because the tattooing either decreases this one or increases this one. So I think that's a very nice point you can take out of that work. Again, I talked about the dose. Now, anatomically as well, if you're really looking at the impact on vaccine responses, well, if you have, for instance, tattoos on your legs and you get vaccines in your arm, it's not going to do much or anything at all. They actually tested this in this paper. I think it's in the supplementary material, where they tattoo, for instance, the left foot and give the vaccine in the right foot, and then there is no effect, right? So it's anatomically specific. So it's again the specific draining lymph node on the anatomical side where you got the tattoo. So if you have nothing in your arms and you get shots in your vaccines in your arms, it's not going to do much. If you have a tattoo on your foot, for instance, a small one. So that's also like you need to recontextualize these results. You can't just like, tattoos, because I think one of the headlines was tattooing damages the immune system or something like that. Of course, like this, it's great. Like that, you're gonna catch some views with that, right? But you need to contextualize this thing. It's very anatomically specific. It's ink specific. So you cannot put everything together as tattooing damages, like whatever, creates inflammation. You cannot do that unless it's just clickbait. So that's it. That's an important aspect. Now, in terms of inflammation, what they call inflammation, as I said previously, inflammation is a very vague word. Now, when I listened to the Dark Horse podcast with Brett Weinstein, they were putting it a bit, again, I don't want to criticize anything. They just mentioned, well, is it something that MAHA should look at, make America healthy again? Is that an additional thing that you need to take into consideration if you're looking at overall health, next to lifestyle, nutrition, all the other stuff, right? Drugs, pharmaceutical drugs. It's very hard to put their information into context. You could look at a very obese individual, take a biopsy of the adipose tissue, um, extract the immune cells of the adipose tissue, and detect massive amounts of inflammation. So it's very hard to say, okay, that amount of cytokine that they see, that increase or immune cell recruitment that you would also get with just injuring the skin. How much of a percentage of damage is that? How much of an inflammation is that? Let's assume if you're obese, you have a lean person that gets tattoos and an obese person. Well, perhaps you can look in the blood, like in a highly obese person, you actually see increased inflammation systemically in the entire blood. You can see it definitely in the adipose tissue, but also in blood. So how much of a red flag is this, right? Should this be considered? If you're thinking about all the choices you're making in your life, should getting no tattoos so that I don't inflame the draining lymph node, how much of a emphasis should that be? Now I am biased, I can't say much. But surely these results, I think there's a signal there. But I think in no case should this paper be the basis of making a strong case, that there's some significantly damaging effects of tattooing on the immune system. That being said, I think this big signal is really how long these inks survive or are being detected in the draining lymph node, and the fact that different inks would be used in different contexts, different countries, different regulations. It's almost like the food dyes that they just now banned in the US, like how many of these things, how many countries would have detected some really harsh toxicity of some of these inks and would have banned them, and other countries would say, well, no, it's fine, they're cheaper, something like that. So I think from a regulatory perspective, there's definitely enough of a signal to regulate these things. That being said, I'm not sure how you would regulate them. What kind of assays or what kind of experiments or tests would you run in terms of toxicity? Will you just do human cell culture and just add different concentrations, and if it reaches a specific amount of cell death for a specific concentration, then you would ban it? I'm not sure exactly how you would test that, but there's definitely a hint here that efforts have to be made to somehow come up with a system to figure out what is too toxic or what is too foreign to the human body to warrant a removal. There are some signals in the literature as well that there might be links to things like the big C, cancer. That would be part of the test you would do, right? Is it cancerogenic? Because if it hangs around for that amount of time, if it's some kind of a dye that intercalates in your DNA or things like that, then definitely that would be a big no-no. Would have to be banned immediately. I would assume that some of these things are already in place, but I don't know for sure. So that's my big concern here: the long-term detection and the potential use of carcinogens in some countries, or depending on the ink that's been used. I think it's immunologically quite interesting. It definitely has long-term consequences. I'm not entirely sure this paper really addresses the consequences or gives me a good indication of how severe or significant the consequences are. The only way to address that would obviously be human studies. I doubt that these would be feasible for the simple reason, like, if you really wanted to address that, you would have to have, well, if you wanted to do the same thing. And I'm not sure, the vaccination is a typical thing you do because it's very reproducible. It takes the minimal effort. The methodology is very simple. So I understand why they addressed the functional consequence of this change they've seen in the draining lymph node via vaccination, because it's easy to do. But is that really what you care about? Whether having tattoos changes your vaccination? I mean, maybe it does. Maybe you are concerned by that. But what I was potentially missing in this paper, and might have perhaps expected to end up in a journal like PNAS, would have been transcriptomic analysis, so RNA-seq of the draining lymph node. Not necessarily single cell RNA-seq, but at least bulk. Because there's so many people who do transcriptomic work, and so as soon as you have transcriptomic work, you can put it into context because you can use data sets from in other settings and compare, okay, well, how much is the gene signature or gene expression differences that what I see with tattooing, how big are they compared to maybe something major, something else, a big injury or something potent. If you have transcriptomic data, you have a better ability to put it into the larger context of how big of a deal is this, and this is definitely lacking. The immune cell recruitment and the cytokine data is not enough to do that. So yeah, I'll leave it at that. Yeah, just to finish with that human trial. Yeah, I would probably not. If a human trial would be done, I would potentially not gear it towards a vaccination, but more towards taking biopsies and seeing, or taking actually the draining lymph node. I mean, again, it would be hard to get ethics for that. And look at do transcriptomic analysis of the draining lymph node. But yeah, sorry, I lost my train of thought. So the reason why it would complicate it to do such a trial is, first of all, you would have to recruit people that are planning to get a tattoo. How would you control? That you cannot exactly ethically tell people this is the tattoo you're gonna get with this ink, this amount of ink, this surface. So you would have to recruit a whole bunch of people so that you can actually compare. Okay, how many people are we gonna get that are gonna get something on their right arm with this amount of square inch covered by the tattoo with this kind of inks? You would have to control so many things so that you can have different groups. And imagine that not only are there differences between ink colors, but also two different black inks or two different red inks based on the origin. So to do a trial like that would be an absolute nightmare. I'm not sure it would actually be feasible. And if you wanted to do it with vaccination, you would have to time it in such a way that, okay, so we need to recruit people that get tattooed in just before 2 months before winter, and then they get their flu shot, and then we can. So it would be so restrictive that I'm not sure you would be able to do something like that. So in a nutshell, and maybe I just want to comfort myself for having inked skin. Uh, I don't think it's that much of a deal. Although the authors in the discussion bring up a few papers that I would categorize as rather anecdotal, if tattooing would be such a big issue, I am fairly certain, although not 100%, that it would already been picked up. We would know by now if, oh yeah, we get all these like people getting tattoos, and then suddenly, they drop dead after 2 years. I'm obviously I'm exaggerating like manyfold, but you know what I'm saying. Like if it would be such an obvious signal, I think it would have been detected already. The fact that it's so diverse, again, different countries, different regulation, different inks, different anatomical sites, different nutrition, different obese metabolic status, obese versus non-obese. Maybe that's what hindered so far the detection of a signal. But yeah, if I had I need to come up with one sentence to give you a take-home message. So my take-home message is, let me think about that. It's something that it's nice to know, and maybe later on I can put it into more context. So it's interesting, it's a signal, but my concern remains fairly low at this stage. That's my take-home message. So MAHA it definitely doesn't reach the threshold by a mile to be picked up by MAHA as another lifestyle factor, some external environmental factor that they should start controlling. I think you're far better off cleaning up your diet, exercising, and if you do that, if you have a clean diet, you exercise, you have a great mental status, you reduce stress, you become spiritual, and you develop that side of yourself. The proportion of your skin being inked should be the least of your worries, I think, if you do all that. That's my take-home message. I hope this was interesting to you. I hope it was something you enjoyed. If you come across any immune-related headlines or things or studies that came out and you would like me to have a look, by all means put it in the comments. I always think these things are fairly fun. I took this one on because it was kind of a personal interest of mine, but again, I've put a reviewer hat on many times in my academic career, so this is something I enjoy doing, and I definitely would do it again in the future. So let me know. All right, take care.